So now we are going to start with the second session of presentations. It's going to start with Holmes from Essex University. For those of you that were here yesterday in the eye tracking workshop, he made already a small presentation and I was going to follow up with that.
Yeah, we'll go into a bit more detail today. Hiya, I'm Luke Holmes. I'm a Ph.D. student from the psychology department. And what I'm going to cover today is basically sort of an overview of how we assess sexual arousal, how you study it, and the kind of problems that arise as a result of the methods that we use and also the equipment as well. So, ideally what we'd like to be able to do is just sort of bring people into the lab and show them a stimulus and say, how arousing did you find that? But there's a lot of reasons that doesn't really work. There's sort of social desirability biases and attitudes towards like LGBT issues and stuff like that. So when you ask people these questions, typically the responses you get aren't particularly accurate. We do still ask anyway, just because it's interesting to compare them to the objective measure. But we do need an objective measure, and the one that we use is genital arousal. So this is I couldn't show it yesterday, but this is our little booth in the lab. So participants sit in this comfy chair here. The door is closed, so it's all done in complete privacy. And whilst they're in there, they watch some quite explicit videos, and we measure their genital arousal in response to it. So I'm just going to show the devices that we use. This is the one that we use for men. It's a penile plethysmograph. Basically, what that is on the end there, that little loop, is a rubber band that's full of liquid metal, and a current is run through it. So it goes about halfway down the penis, and as they become aroused, obviously it gets stretched, which increases the resistance on the circuit, and that's sort of registered on the computer as an uptick in a graph. Then we've got the vaginal photoplethysmograph that we use for women, and as you can see, it's about the size of a tampon. It's inserted into the vagina up to that rubber stopper that you can see. I think it's about 2.5 inches. And the way that one works is it contains a red LED and a photoreceptor. As blood is pumped to the vaginal walls during arousal, they become darker, so the level of sort of reflected red light changes. Now, as you can see, they're both named plethysmograph, which is literally just an instrument for measuring changes in volume within an organ. But you can already see a sort of fundamental difference here, which is that normally with a plethysmograph, it's used on like a muscle or a lung, and it's just a rubber band, and so the changes in volume that we're measuring are persistent ones. So it will stay there. Whereas with this, we're measuring blood that's being pumped in, but then it's being pumped out again. So there's a sort of fundamental difference in the nature of the two devices. So this is the kind of results that we gather. This is a 40-minute experiment here with a straight male. We display 6 sexual videos during the experiment, all of which are roughly similar in content. So they feature a lone model in a room masturbating. And they've been chosen during a pilot study for their attractiveness. So we know that these people do work. And as you can see, he responds only to the females and nothing to the males whatsoever. With a female participant, this is the kind of result we get. I've zoomed all the way in. This isn't the full 40 minutes. But as you can see, we're essentially measuring a pulse rate. We're measuring a heartbeat. So there's no stimulus here, and then the stimulus begins somewhere here, and you can see that the amplitude increases. So more blood is being pumped down there and then pumped away. But when we zoom out to look at the full 40 minutes, you see an interesting pattern, which is that despite this participant being straight, she displays arousal to the females as well as the males. And statistically, there's no difference between females and males for this participant. This device to measure arousal in women was invented a few decades after the male one. And at first it seemed to be working just fine when they were showing sort of heterosexual, just a man and a woman having sex, and they were showing arousal. When they had the bright idea to split it into males and females separately, they suddenly realized that they were showing responses to everything. And so they had this question of, is this an artifact of the device? Is the device that we've invented to measure this just completely faulty? And are we really measuring arousal at all? So this is how our results look when you aggregate them. This is a graph showing all of the males that I've ever tested in the lab. Sexual orientation is on the x-axis, and we've got our straight males here at the 0, and the exclusively gay males at the 6s here. And in the middle, you've got sort of varying degrees of bisexuality. Now, on the y-axis, what we've done is we take their arousal to males and we deduct from that their arousal to females. So if you respond much more to females than males, you end up with a negative score, you end up below this 0. And if you respond more to the males, then you end up all the way up there. And as you can see, it pretty much is, the line is a regression line with a 95% confidence intervals as well. And this pattern is more or less exactly what you'd expect it to be, where the straight guys are only responding to women and the gay guys are only responding to men. But when you aggregate women together, yeah, there we go, there's a huge amount of variance across the entire spectrum, but also the confidence intervals for the straight women include 0. And that means that actually they're not showing a preference in either direction. And so we have this problem of, is it down to the device? Why have we got this strange pattern of results? And is this really like a sex difference between men and women? Is there a fundamental difference in how their arousal works? But that's not the only problem that we have. We also have another one, which is maybe we're subject to volunteer bias. Perhaps the participants who are willing to come in and have their genitals probed in our lab have strange properties that the general population doesn't have. I'm sure there's people in the audience right now who are thinking, I would never volunteer for this. We pay very well, but unfortunately this has been studied, and the reality is we do have a volunteer bias as well, especially when it comes to experiments involving genitals, and especially in women. You can see here that the volunteers were found to have greater non-coital sexual experience, greater masturbatory experience, less sexual inhibition, and more experience with what they call unusual sex. So we're potentially testing a sort of odd subsample of people and are subject to volunteer bias here. So what we do to sort of alleviate this is use pupil dilation tracking as well. So you see here we have this sort of mobile metal table. This is an eye tracker just like the one that we looked at in the demo yesterday, so it's an incredibly basic one. We use that to measure their pupil dilation whilst they watch the videos in addition to their genital arousal. So it's already been established that eyes and genitals do correspond with each other, that they correlate together, and that we get the same sex difference and sexual orientation difference as we do with genital arousal. Pupil dilation works as a measure of this because it reflects activation of the autonomic nervous system, so the sort of all of the automatic processes that you have, like breathing, reflex actions, and sort of anything that you don't really have to think about, anything that doesn't involve the brain, is controlled by this system. And as a result, participants can't really control it. They can sort of exert will and control their genital response to some degree, but that's much more difficult to do with pupil dilation. Immediately after you've displayed a stimulus, they'll show a response typically within sort of milliseconds, so it's a highly responsive measure as well. Fortunately for us as well, it seems that pupil dilation to the preferred sex might be the strongest pupillary response which exists. There are plenty of different things that make your pupils dilate. Just being interested in something will cause it to dilate, but fortunately for us, it seems that we're dealing with an effect that's quite large in magnitude. Crucially, it provides the same measure for men and women, so we can suddenly compare their results directly. So I've got the exact same data set as before here, but this is for pupil dilation. You can see that with men you get roughly the same pattern. We've sort of lost it. We've dropped from a regression coefficient of 0.9 to 0.6, so this line is less steep than it was. And for women you get the exact same result as you would with genital arousal. And I've put these next to each other just so that we can sort of flip between them. You see you get a much less extreme response, but you still see this lack of specificity in women. But another problem that we have is that explicit sexual stimuli is still invasive, so there's still people who won't come in if you're using that kind of stimulus in your lab. And so we decided to test whether people dilate to non-explicit stimuli as well, and this is what I showed in the demonstration in the lab yesterday. So we took 165 men and 160 women, and we made sure that we had a nice distribution across the Kinsey scale, which means that we have a mix of different sexual orientations. And in total, we had 48 stimuli, all of which were 30 seconds long. The explicit ones were just models masturbating, as we always use, and the non-explicit ones were basically attractive students talking about the weather. So these people have been selected for the fact that they're physically attractive, but there's nothing sexual going on, and obviously they have clothes on and everything like that. And we measured both pupil dilation and viewing time as well. So the first prediction we had is that we would expect that the dilation to the non-explicit stimuli would correspond to the dilation to the explicit stimuli. So in other words, if a person dilates very strongly to women, we would expect them to dilate strongly both to non-explicit and explicit depictions of women. So in men, you see that we get so all four of them were significant correlations, sort of weak to moderate correlations on these, but we did find in between sexes and across sexual orientations as well, we found that their dilation to men and women sort of corresponded with each other. So in other words, we're looking at something that's measuring the same thing as the explicit ones. And for prediction 2, we sort of threw all of our measures together. We correlated pupil dilation, viewing time, which I'll show you in a minute, and the self-reported attraction, so the sort of subjective scores we have for how attractive someone is. And hopefully this will work. It worked during the demo, so we'll see. Yes, here we go. So this is our viewing time section. These are two students that have been selected as being particularly attractive, and all we've done is put them next to each other. This is the kind of experiment, the kind of paradigm that I wouldn't expect to work, but it does in fact work incredibly well. All you have to do is sum up the amount of milliseconds people were looking at either the male or the female, and what you'll find is that men show a very strong preference. They just sit and stare at the woman the entire time, and women sort of flick back and forth and end up not showing any kind of preference in any direction. So we find the exact same pattern with this as well. And when we correlated these together, we did it separately for the explicit and the non-explicit stuff, just so that we could compare between the two. So with the explicit stimuli, we had an average correlation between these three, we've sort of averaged the figure, of 0.64 for men, so they corresponded quite nicely, and 0.53 for women, and this difference between men and women was significant. And then for the non-explicit, we found a weaker relationship, so we're down to 0.47 from 0.64 for men, and we're down to 0.41 from 0.53 for women, but they were still highly significant themselves, and also the sex difference was significant too. So it seems that you can use non-explicit stuff and still get roughly the same patterns that you would. There was another paper a few years later which sort of disagreed with these results, but I was still quite happy about it anyway, because what they did was took stimuli where they had the exact same person in the exact same pose, either with clothes on or without clothes on, and they found that there was no difference. So in other words, if you use non-explicit stimuli, you don't get a weaker relationship, or at least that's what they found with pupil dilation. The one issue is that they used porn stars for this, so all of the people that they used were really attractive and have some quite pronounced sexual features, secondary sexual characteristics. So I don't know if this will work as well with the average person, but it's a good sign anyway. So it looks as if the sex difference that we see in genital response does in fact reflect a genuine difference between men and women on some kind of biological level, and it isn't a measurement artifact just based on the fact that we're using a different device to measure genital arousal. And the use of pupil dilation is a particularly interesting one, because if we use that along with the non-explicit stimuli, we could potentially reach people who wouldn't otherwise take part. And that doesn't just mean people who on campus who won't take part, but there are whole areas of the world where this kind of research has never been conducted because people just aren't really up for the procedure. So potentially we could go to places that we wouldn't otherwise be able to study and use just pupil dilation alone with clothed models and maybe get some interesting results, and we can see if our results are generalizable. And yeah, that's it. Thank you very much. Stunned into silence. No, there's
Yeah, I have a question about the methodology you use. You had 24 videos.
Yeah.
One with sexual content and 24 without or explicit content.
That's right.
So
yeah.
it was 24 in a row of the same, or they were mixed?
No, they were mixed. Well, it depends on the specific. There were several blocks that were slightly different conditions, but they were mixed together. They were randomized in terms of the order of the blocks and the order of the videos as well. Because people get a bit worn out if you show them loads of explicit stuff in a row. They just sort of. So what we do normally in our experiment is we show people 3 minutes of porn, and then we ask them how attractive they found the person and how arousing they found it and stuff like that. And then we show them 2 minutes of David Attenborough just to take their mind off what's going on. So we did something similar there. It's been a while, so I can't remember the specifics, but yeah, it was all randomised.
Maybe that voice also on, because that maybe...
It's the nature, yeah. You can't see David Attenborough himself. You can just see animals. Unfortunately, no one has responded to that yet. No.
No, but what I was wondering is maybe you may have a post effect after seeing this explicit content. Seeing, even if it's not explicit, you may more. For example, you say that males focus more on females, but females don't focus so much on males. That's the general one, I assume, but is there a difference, for example, for the video next to a explicit one?
Yeah, it gets a bit tricky with this. Fortunately, with pupil dilation, we don't have to deal with this as much because people return to baseline almost immediately after the stimulus is gone. So we're very grateful for that. Normally, we have sort of a long period where we have to be careful with how we compare the results. So if we show someone porn and we're doing genital arousal, we'll compare their response to that versus the 10 seconds immediately before the video. So even if someone is still sort of partially aroused, because we're comparing it to the beginning section of the video, we can control for sort of changes in volume that persist a little bit. But that's the amazing thing about pupil dilation, is with the whole thing being so quick, you can literally show a person a stimulus, their pupils will dilate within a second, and then as soon as you end the stimulus, they return back to where they were, which is amazingly convenient. There's no waiting around or making them do counting tasks over the intercom or anything like that.
Question about, so with genital arousal, I have read this theory that women might be responding because they feel empathy towards the women they're watching. And would this theory then predict the same sort of pattern in pupil dilation, because it seems to be a bit less related, perhaps?
Yeah, that's a tricky one. There's loads of theories about why women might have this non-specific pattern, because this has been found not just in genital arousal and pupil dilation, but in all sorts of brain imaging studies and just everything. There seems to just be no specificity whatsoever on a group level. The empathy thing is a sort of a worse explanation for pupil dilation than it is for genital arousal. But unfortunately, no one has any idea why this is, and all the theories that people come up with end up being disproven by an experiment like 1 year later. So 2 years ago now, there was a paper that got published that didn't have any form of experiment in it. It was just 10 competing theories trying to explain the same thing, and I'm pretty sure over half of them have already been sort of disproven with another experiment. Yeah. So no one knows. If anyone has any ideas, I'm happy to listen. That's it. Yeah, thanks.
Thank you very much.